All right, so we know that enzymes are very large, complex proteins. We know they speed up chemical reactions, they lower the activation energy, and we talked about carbonic anhydrase, which makes it possible for us to not die from carbon dioxide building up in our cells. But how does an enzyme, such as carbonic anhydrase, actually work? So we need to understand something about chemical reactions and the structure of enzymes. Chemical reactions have what we call reactants and products. And when we're talking about enzymes, oftentimes the reactants are known as substrates. That's a new word. So substrate, reactant, same kind of idea. And what happens is an enzyme has what's known as an active site. Okay, so that's a very important term right there. And the active site is the place where the substrates or the reactants can actually come together and meet. So right here in this model here, showing all of the molecules in an enzyme, is the active site. Doesn't look like much, so we don't, at this level, look at it this way. Here is another drawing of an enzyme. So this yellow blob is the enzyme. This right here is our substrate. Here's the active site. Okay, so that substrate can fit into the active site just like a key fits into a lock. Now, each enzyme has a very specific shape, okay? And the active site is very, very specific, and it has to do with bonding and all sorts of stuff. And so that means each enzyme works for only one specific type of reaction, and it depends on the active site, which substrate will fit into there. If we focus on this picture here for a moment, okay, so now our enzyme is purple. Here we have two substrates. So let's say we have carbon dioxide and water. That they're going to come together to form carbonic acid, soluble in your blood. So right here, this enzyme would be carbonic anhydrase. So you can see how the two substrates would fit perfectly into the active site. Right here. This is known as the enzyme-substrate complex. What happens is while the substrates, so in this case, we're putting two molecules together. It's known as a synthesis reaction. What happens is when the carbon dioxide and the water fit into the active site of the enzyme basically helps to form the bonds between those two molecules. Then you get over here the carbonic acid. Okay? That's our product. Notice that the enzyme itself looks exactly the same as it did here. So the enzyme is unchanged. Okay? So it's unchanged, which means it can be used again and again and again and again and again. That is very important. So we have lots of different enzymes in our bodies, but they all can be used repeatedly. There are a couple of things that can make enzymes not work as well. These factors that can affect enzymes are temperature and pH. So what temperature can do is basically as temperature increases, enzymes tend to work faster because, as you hopefully know, molecules move around more quickly at higher temperatures. That just means that the substrates fit into the enzyme's active site more quickly and the reaction occurs that much faster. But that only happens up to a certain point. Okay, so up to a certain point. Basically, if you boil an enzyme, for example, if you increase the temperature too much, it's not going to work anymore. pH can also have an effect on enzymes. Some enzymes work best at neutral pH, around pH of 7. Other enzymes might work best at a pH that's very acidic, like the enzymes that are found in our stomach because of all the stomach acid there. If an enzyme does not work anymore, due to maybe the temperature being too high or the pH being changed too much, too acidic or too basic, then we use the term denatured. So we say the enzyme has been denatured. What that means is the shape of the active site has changed, so then the substrates don't fit in there anymore. So we'll look at an example of how that can happen in class. So that, in a nutshell, is how enzymes work.